for inviting me to come and give a seminar. I was just saying earlier that you have all brought me some very good luck, and there is finally some sunshine in what has been a very rainy start of the summer here in the UK. So hopefully over the next 40 minutes or so, I will get an opportunity to speak a little bit about my own research, focusing around some of the challenges that we see in stem cell bioprocessing, and sort of that age-old question in engineering of whether we should be scaling up or scaling out, and what are some of the particular challenges associated with those choices. But as has just been mentioned, I'll also give you a little bit of a flavour of some of the work that we do within the school that I'm part of in Loughborough University. So really, we are a school which has about 85 academics, and for those of you that don't quite know where Loughborough is, essentially we are based sort of in the middle of the UK. If anyone is a fan of Sherwood Forest, Robin Hood, that is just based up around sort of Nottingham area, and we are just here. Unfortunately, we don't have a particularly famous football club to be associated with, so we're not particularly well known. But we have a very large campus, and we're based in engineering, sort of at this end. Our S building, as we know, where chemical engineering space, for example, is just over here. As I said, we're sort of a reasonably sized school. We have about 1,200 of us in total, made up of a range of MSc students, PhD researchers, sort of RAs, academic staff. As I said, about 85 of us in total, slightly more in aeronautical and automotive engineering than in chemical and materials. We have a good number of support staff and some that focus a little bit more just on teaching. And we are indeed made up of those three different departments that you can see just there: aeronautical and automotive engineering, chemical engineering, and materials as well. And if we look at what some of our school research priorities or themes are, healthcare features quite heavily in there. So we do a range of work across bioengineering, thinking about biomaterials as well. Some of that is not always focused around healthcare, but also about how we can use biomaterials in other ways, particularly thinking about things like bioremediation, water treatment, etc. The sort of regenerative medicine work that I do, pharma manufacture as well, and focusing around antimicrobial resistance. And that interfaces quite nicely with what you can see here on the side of the slide, which is our university level sort of strategic themes, focusing around sport, health and wellbeing, climate change and net zero, and also vibrant and inclusive communities. So I'm just going to talk about four researchers other than myself. Again, just a slide on each to give you a little bit of a snapshot before I delve into my own work a little bit more deeply. And the first of these is Professor Danish Malik, and he's really been focusing over the last few years on bacteriophage production and storage. So bacteriophage, for those of you that don't know about these, are effectively viruses that can attack bacteria. And so as part of the antimicrobial resistance movement, where people are looking at new ways to tackle bacteria, because we know that our current antibiotics are no longer as effective against certain strains because of the resistance that they have developed. These bacteriophage, which evolve alongside the bacteria and are highly specific to each bacterial strain they can infect, are potentially a really good option to use therapeutically, and also within potentially animal feed, for example, because we know that actually a lot of our sort of problems, if you like, with antimicrobial resistance comes from the overuse of antibiotics in things like chicken feed, raising pigs, cows, etc. So what Danish has been doing is really thinking as a chemical engineer about how we can tackle that problem with that engineering head on. So rather than thinking about how we identify the correct bacteriophage, he's been tackling the issue of actually how do we grow enough bacteriophage to potentially be useful in those industries, and also thinking about how we can store them, how we can actually treat these bacteriophage so that we can then incorporate them into things like animal food, because we know that some of the processes that are associated with the making of animal feed include a lot of high temperatures and so forth. So what you can see here on the slide is this idea that actually is creating what he calls a core shell capsule, one material which is directly surrounding bacteriophage, and then another material that encapsulates that whole mixture so that it's protected by this core and shell. Okay, let me have a little nosy. So I don't seem to have many options to do with my microphone other than turning it off and on. Let me just have a look. Right, is that any better? Okay, right. We'll stick with that then, but shout if it's a problem. So I was saying these bacteriophages, these viruses against bacteria, they've now been encapsulated in this core shell, and you can see what that looks like just here. They're all inside this white part of the particle, and then that's a sort of shell. And that means that actually we can release these bacteriophage in a pH responsive manner. And if we think about our own digestive systems, but also those of animals, you will have therefore the ability to regulate whether those bacteriophage are released in the stomach, small intestine, and so forth. And also, he's been showing that having that protective capsule around the bacteriophage also means that actually you can they can survive for a short amount of time those really high temperatures that have come along with things like extrusion processing to make pelleted animal food. So that's what Danish has been working on. We then have, mouse is willing to play ball, there we go. Elizabeth Ratcliffe, who's in the department, and again, she's very much thinking about bioprocess optimization. So she has worked on a range of different product types. So here again, this is some work that she happens to have done with bacteriophage. And really what she's been doing is showing that you can use a scaled down shake flask model to look at whether those process variables impact phage production. So for example, looking at things like the temperature of the culture when you actually infect the bacteria to grow the bacteriophage when you're trying to scale up their culture, and also showing, for example, that scale down model gives you the same type of results as then a 5-liter bioreactor, showing that the changes that she was making to the process are compatible with then a scale up process. And so you can see that sort of you have an initial sort of before something has been changed to the process, and then once she's optimized the conditions, the type of increase in productivity that she can achieve. And she's done this sort of processing with viral vectors as well, for instance. Moving away from bacteriophage, we have, for example, Dr. Aslam Islam, who's really focused on metabolic engineering. So this is just some work where he was generating ethylene glycol, which is used as in the manufacture of various commodity chemicals, but wanting to do so via biological route. And so what he was doing here is describing a novel synthetic biochemical pathway and a particular bacterium, a particular Clostridium strain, where they're wanting to convert acetate into ethylene glycol. And so he had two parts to this work. One where he was very much focused on doing the computational side of the work, where he was developing workflows that helped him design novel biochemical pathways. And then a second part where we actually then, he was introducing through genetic modification, those new pathways into the Clostridium strain and looking at whether or not we could actually generate enough ethylene glycol. As you can see from what he is showing here, what I've circled is that actually the yield of the ethylene glycol was fairly low, but this was really a proof of concept piece of work for him to show that he could do this combination of computational work to design an ideal sort of system and then actually translate that into the real world in a bacterial system and get a result. So now he's going to go through lots of optimization to actually increase that yield. Again, a slightly different spin as well, sort of leading more into some of the pharmaceutical type of work that we do, which is also a lot on the digital engineering front and looking at sort of digital twinning and things like that for the pharmaceutical industry. We have Kuai-Yu Yang, who's really focused on protein crystallization. And what he's particularly interested in with protein crystallization is not just being able to look at protein structure, but actually thinking about this as a potential tool to help in protein not just synthesis, but actually downstream processing. So looking at using it for purification. So what he was doing here is really investigating whether, for example, using graphite or graphene oxide as a template for some of the catalysis, using lysozyme as a model protein, and trying to understand how you make some of those informed choices about what structures to use for your crystallization. And so he's finding, for example, that using the templates, you could change the crystal morphology, you could change the crystallization kinetics, and that's just what this graph that I've picked here from the paper is showing. So a quite wide range of things in the sort of bioengineering space that we do at Loughborough University, and always happy to have a chat if you have any sort of ideas for collaboration, etc., to try and put you in touch with the right people at this end. But moving into my own work, as I said, I'm going to talk really about some of the challenges around stem cell bioprocessing. And so when I started working in the field just about 15, 16 years ago, our goal in the kind of cell therapy bioprocessing world was to make sure that we can generate cells, with a reliable therapeutic function, which have regulatorily acceptable safety profile to make sure that there's no other cells, for example, in that dose, there's no sort of danger to the patient. But we need to do so at what we call clinically relevant numbers to make sure there actually would be an efficacy. And of course, at a cost, which, depending on the type of healthcare system that you're working in, allows for reimbursement through perhaps insurance, or allows you to actually fund that through something like our National Health Service in the UK. And when I sort of, was looking at that goal that the industry had set for itself, I also thought to myself, well, actually, really, that's also our challenge in this sector. It's not just the goal, but equally, all of those things are quite difficult to achieve individually. They're even harder to achieve altogether. And so the cell type that myself and a colleague of mine, Chris Hewitt, and others sort of decided to focus on was actually human mesenchymal stem cells. And so these are really interesting cells to work with because they can be isolated from various sources. Typically, we work with those that have been isolated from bone marrow, but they're quite rare within the bone marrow, so we have to work quite hard at isolating them. One of the reasons we're really interested in these stem cells is that they're able to differentiate into osteoblasts, chondrocytes, and adipocytes in vitro. And actually, in laboratory settings, lots of researchers have been able to show that if you can manipulate them through either small molecules, but also surface topography, etc., that you can turn them into cells other than these. But typically from the bone marrow in the human body, this is what they would focus on making. There's a lot of debate about actually what defines a mesenchymal stem cell, and there's lots of different terminology like mesenchymal stromal cells, stromal stem cells, etc. out there. And quite a long time ago now, they were defined by a seminal paper as having these particular sets of markers. And that's really reflective of the fact that in the bone marrow, we would typically also have hematopoietic stem cells that make all of our blood cells. And so what we're really doing with this set of markers is defining that they are not hematopoietic stem cells, they are other. And so there's still a little bit of debate about if you isolate these from, for example, fat pads or from other areas of the body, whether they truly are the same stem cells. But this is how we've been defining our particular mesenchymal stem cells for a long time now. One of the questions I often get asked is, if they're quite rare, these cells, and actually they're a little bit undefined because you have this range of markers, you don't have a single marker that tells you you're working with the right cells, why are they a pretty good sort of therapeutic option? Why are we interested in them? And this is some data that one of our students pulled out, nearly a decade ago now, but actually the numbers are pretty much still the same if you rerun them now. But really it's interesting because compared to what at the time, 15 years ago, was a growing area of interest in embryonic stem cells, which came with lots of ethical concerns, these are much less ethically contentious because although I would never say that a bone marrow sample would be painless to have, it doesn't come with some of those ethical implications of working on embryonic stem cells. It's also really interesting because MSCs themselves are the therapeutic cell type, so we don't need to worry about having to turn them into something else for them to function. And therefore, it means that we can do also autologous therapy, where we give the cells back to the same patient, or allogeneic therapy, where we can give them to lots of different patients because they have this odd sort of way of getting around the immune system. So they don't tend to be detected by the immune system and means they're a really interesting option for us to work with. And you can see from that data that our student have pulled together from clinical trials databases that, there was a lot of work happening with mesenchymal stem cells, and they were pretty much across the sort of range of the clinical spectrum. We know, of course, by now that the CAR T cell therapies have really beaten mesenchymal stem cells in terms of being those first high-level clinical successes and talked about an awful lot in the media. But one of the challenges of working with mesenchymal stem cells, and in fairness, sort of any cell therapy, is that we do need to generate quite high numbers of cells in order for them to be therapeutically functional. So again, this is about a decade old now, but if you look at the more recent literature, you're seeing a lot of the same numbers crop up, that we often need about 20 million, 200 million cells per patient or per kilogram of body weight, depending on what measure a particular clinical trial was looking at. And you can see here that, again, if you look at other cell types in the sort of stem cell therapy world, we're talking about quite similar numbers. So if anyone in the audience has ever grown cells in a tea flask, you will appreciate, depending on the tea flask surface area, because these are adherent cells, you might get several million within a single flask. But to get to 200 million for a single patient, we're talking about a lot of surface and therefore a lot of space in an incubator, etc. So if we look at sort of what bioprocessing for these human mesenchymal stem cells would look like, really we're talking about having to have a way of taking the sample from a donor or potentially the patient themselves, some form of initial growth. We go into a scale-up system. We might then bank these cells down to make sure that we have enough of a working cell bank or production cell bank if we're talking about allogeneic therapies. We then might go to an even larger scale. We then have to harvest the cells and potentially concentrate them. We may or may not introduce a cell separation step. At the moment, we still very much tend to give a bulk of mesenchymal stem cells. We haven't yet as an industry identified whether there are certain ones within the population that are more or less efficacious, but that could come in the future. And we go to some form of formulation and putting these into the patient. And so there's lots of challenges along that whole pathway. As soon as you work with any human cells and donor material, we know that there can be lots of variability in that input material. It's a very complex regulatory framework that we're working within. There's some challenges around how do we create shelf life, and actually that's one of the things that my team has really decided to focus on. And one of the reasons that we wanted to focus on sort of cell storage is because we decided in the great debate of scale up versus scale out, particularly for allogeneic therapies, that we were going to take a scale up approach. And that meant that we might have very large volumes of cells that we then need to store at some point in the process. So I'm just going to talk you through for a little bit kind of what our reasoning was in terms of making that scale up versus scale out decision. And so when we looked at scale out, being in a department of chemical engineering, it won't surprise you that we thought about stir tank bioreactors. When we really recognized that that was going to involve a transformation of a process that was very based on tea flasks in most laboratories. It could be low cost, but high risk potentially. There's a high potential, reward. It could take a long time to market, but there's some advantages because actually the regulators sort of know and love stir tanks, and we can build, systems around them in terms of what we can measure about the process to ensure quality. And we sort of ended up identifying that that was going to be more suited probably to those allogeneic therapies where we're taking cells from a single donor and potentially treating lots of patients. And the reason we went down that sort of route is because when we looked at some of the scale out options, for example, whether we go into sort of cell factory systems, we had some concerns there because the stem cells are the product. And so we were quite concerned at the time that having, some of the gradients that we do see in cell factory systems of either oxygen or nutrients, that it would mean we end up with quite a heterogeneous population of stem cells, and that wouldn't necessarily therefore meet regulatory standards in terms of quality of product. So we decided to sort of not go down that route. And we did consider more using automation. So we actually did some work with a company called the Automation Partnership, which then was bought out by Sartorius eventually. But they had developed this equipment that you can see here called the Compact Select. And that's basically a robot doing what a human would do with a tea flask. So it can read a barcode on the tea flask, identify which patient those cells belong to. It will remove the medium. It will add trypsin, for example, to then remove the cells from that flask. It will collate them, send them for centrifugation. You can then plate out into new flasks, etc. And so this is something that we considered. The nice thing is that you're transferring sort of an existing manual process into automation, so it's a little bit less risky in that sense, but this equipment is very expensive. And it might therefore have a shorter time to market because we have an existing manual process. But ultimately, again, because of the sheer amount of cubic sort of centimeters squared of surface area that you would need to create those 200 million cells per patient, this became quite untenable. So we decided that actually the automated platforms might be more suited if you were doing an autologous type of therapy, where you're just having to worry about, a patient at a time. And so work that we didn't necessarily do, but that was done in the wider sort of group at Loughborough in what we call our Centre for Biological Engineering. So particularly Rob Thomas and Yang Liu here. They did a bunch of work in that automated platform, the Compact Select, and for example, showed that you absolutely can culture mesenchymal stem cell populations in here. And so, for example, Rob and his team showed that you get actually not quite the same performance in terms of cell yield with the automated system as you do with the manual, but it's pretty good. And there seems to be a slight shift in terms of being able to get higher cell yield at later passages, which was interesting. So Yang then did a piece of work to try and understand why that might be, because there's a lot of interest at the time in how we can use these automated platforms for different cell types. So she did some work using a osteoblast cell line, so a cancer cell line called HOSTE85. And she showed that actually if you had a number of well-trained RAs who were used to working in cell culture and you had them culture cells over a number of passages, despite everyone's best attempt at doing that training and faithfully following protocols, you had quite a wide distribution at the end in terms of the number of cells that they would generate over that set number of passages. If you had the automated Compact Select do this work for you, you had a much, much tighter distribution, but you ended up with fewer cells. So we actually went ahead and did a little bit of extra work with one of our PhD students. And what she was showing is that one of the downsides to the automated machine is that it works quite slowly. So what we were finding is that these mesenchymal stem cells are actually quite sticky. And so what was happening is that in the time it was taking for the robotic arm to be moving around, selecting fresh medium, adding the trypsin, etc., that cells were just often re-sticking to different parts of either the flask, the pipette, and so forth. And that's why we're re-losing some of that volume. But as you might expect from an automated system, it did the same thing time and time again. So you do get this much tighter distribution. So that's just a little bit of a sort of story about how we made, some of that decision and why we went for the scale-up route, really. And so what I want to do now for what I think is about the last sort of 20 minutes of the talk is a little bit more of the sort of story of the scale-up that we did then do. So we decided that really what we needed was a holistic approach. So as I said, I worked with a colleague called Chris Hewitt, and he was very sort of strong in terms of believing that one of the challenges that bioprocessing had faced for a long time, and he was using the monoclonal antibody sort of world as an example, that there was this real throwing it over the wall approach. So you had an upstream team who was doing their work in terms of vector development and plasma development and making sure that your CHO cells or E. coli were producing as much product as possible. And then, they made changes and just went, well, the downstream team will now have to cope with the changes that we've made. And there was never this really joined up thinking done in the sort of early days of monoclonal antibody production, et cetera, et cetera. So we said, we're going into a new field of this regenerative medicine, cell therapies, we should take a different approach. So we tried to look at actually how different parts of the process tie together. So because I was interested in this question of storage, and quite often, as you'll see from the next couple of slides, our main option for storing these stem cell therapies is to freeze them. I really wanted to understand how different choices we were making, whether that's in our actual cryopreservation protocols, or whether that's in, for example, our upstream cell culture process decisions, or in some of the measurement systems that we use, how all of those choices impact on what we then deem to be our cryopreservation success. So how many cells that we can thaw at the end of a process, potentially to go into a patient, that are functional, alive, etc. So where did it all start? Well, as I said, I've been sort of in the field for a little while now, and so back in about 2012, this paper was published, which really highlighted what we all already knew, and it basically said that if you look at the mesenchymal stem cell based sort of clinical trials and things that are going through to the FDA, all of them, more than 80%, are actually looking at this freezing cryopreservation to store and transport the final product, rather than using either fresh cells or cells that are refrigerated, like we do, for example, blood products. The most widely used cryoprotectant, because freezing cells does come with some damage, and that comes from the ice crystal formation, both in terms of physical damage that ice crystals can cause within the cell, but also the osmotic imbalance that happens as you get the pure water freezing inside or outside the cell, causing an osmotic shift. So people use these cryoprotectants, and DMSO is one of the most widely used. It's in most of the cryopreservation sort of solutions that you can buy commercially, and it's used very widely across labs. And that DMSO is needed because it will sort of avoid those osmotic transients during freezing. It changes at what temperature you get some of that cellular ice crystal formation, and so avoid hopefully some of the biochemical damage that we see. The downside is that as many papers that talk about sort of DMSO being really good, and we have been using it for quite a few decades, particularly in the sort of cancer cell storage and banking. There's also lots of reports about how DMSO can be cytotoxic, particularly in a time and dose dependent manner and at temperatures above freezing. It might alter sort of cell function. There's some regulatory concern being aired around this time around DMSO linked side effects, which were quite rare, but with the view to the fact that cell therapy, if it becomes doable and more popular, actually this could be a larger percentage of patients that could experience these. And so it was also quite dependent on the cell type, the disease indication, concentration, whether they were washing cells or not, etc. So that was sort of the world we were working in when we started some of these projects. The other thing that we realized when I had one of my PhD students, Tim, really start to look at cryopreservation as a process and how we might start to maybe go away from some of that use of DMSO potentially, or at least understand more what the damage is. When he was looking up protocols for freezing cells, and these were ones that I myself, apologies, had used when I was working in the laboratory, you quite quickly realize that actually they're quite vague. They say things like work quickly, doesn't say what quickly means, again says work quickly, avoid sort of having them in DMSO for an extended period, but doesn't specify how. And for somebody who, as was mentioned earlier, started off as a pharmacologist and working very much in biological labs, using terminology like happy cells, etc., when I was looking down at a flask of cells, now that I was in this engineering environment and I was being strongly encouraged to think about how do we set boundaries, how do we set kind of limits for our experimental systems, and how do we quantify some of these things, it just made me a little bit uneasy to work with these quite vague processes. So one of the first things that Tim did was actually looking at how we can define a processing window for working with DMSO, particularly thinking about if we're going to scale up, how can we do that working quickly, so to speak. Because actually, as I'm sure you'll know, if you've got PhD students in the lab and if they're ever freezing any sort of cellular material, they can probably, label aliquot and store on ice and then put in the freezer, maybe 20 aliquots in a matter of minutes. But if we're looking at some of the volumes that we might need to generate where we have, trillions of cells in a particular batch, if it's going to take even an automated vial filler an hour, perhaps, to actually aliquot these cells in DMSO and put them into a cold chain, how much damage are we going to see in that case? So Tim set up some experiments where he was really trying to find that tolerance limit about how long cells could be in a DMSO-based suspension at room temperature, assuming that that would be the sort of manufacturing space that would be available.
Muy buenas.
So we exposed our human bone marrow-derived MSCs to 10% DMSO in freezing medium. for either 2 hours sort of on its own, what we call the just exposure, by exposing them to DMSO for up to 2 hours, but then freezing them, or also by potentially freezing them and then exposing them afterwards. And this was less about the manufacturing process and more about actually what would happen at the clinical site. You know, is there a chance that somebody would be ready to start an infusion? There's a distraction, an emergency patient comes in, etc. And at what point do you have to say those cells have now been thawed for too long? We can't use them anymore because of the damage DMSO might be doing. So again, where is that tolerance? And so I'm not quite sure why my red bar has gone flying across the screen here, but 70% viability, according to the FDA, was a sort of standard release criteria. So we use that as a bit of a benchmark. And so you can see that actually, you know, DMSO exposure per se on its own or prior to freezing has relatively little impact on cell viability as measured by membrane integrity, but does start after an hour to actually affect attachment efficiency, as you can see in graph D here. And we start to see, for example, if they've been exposed for longer, the sort of fold expansion, so that ability to then reattach, grow long-term is affected. And actually, it also showed, you know, quite a drastic reduction in that sort of CD marker expression, indicating that the identity, something about that is changing. And even if we don't think it's truly identity, but it's showing this sort of damage to the proteins on the outside of the cell that are picked up in these assays, that's showing us that there's been some kind of damage. So we concluded at this point, quite early on kind of in our work, that any exposure would need to be limited to less than an hour, but it may depend on the cell type, and you need to think carefully about when and what types of assays you choose to measure cell recovery by, because if we had only focused on cell viability, really most of our conditions would have been, you know, absolutely fine, and we wouldn't have picked up on some of this longer term damage. And again, you know, we sort of started to therefore hint at this idea that longer term damage could be picked up on. And again, in the literature, people started to talk about this phrase of delayed onset apoptosis. And it was really starting to highlight to us that although cell recovery might be high immediately post-thaw, there's that damage that takes a little bit longer to become apparent. So again, this is work done by a different student more recently, looking again at adhesion as an example, and the difference between in the sort of pale bars, some fresh samples, and each of these is a different donor of cells. That at 1 hour, you can't really tell the difference between the cell adhesion between our fresh cells that are just in continuous culture versus those that have been frozen and are now thawed. But as soon as you start to look at it a little bit later on in the process, that difference between fresh and frozen becomes quite acute. So, you know, it's again a question of, even at 4 hours, can we tell whether or not these cells are good enough to go back to the patient, for example, because we know they're not behaving quite as normal. So we know actually by now that it's not only the choice of measurement that we need to be really careful about making, but also at what point the measurement you're actually taking. So is it appropriate to do it immediately post-thaw, as is quite typical in sort of release studies when we want to give the cells to the patient and we don't want things sitting around for hours? Or actually, would we be better off holding fire, doing some of this testing and really understanding whether our cells are good enough to go to the patient? Will they work? Something else that we started to look at was whether or not we could reduce the amount of DMSO, because as I said, lots of people were sort of worried about using DMSO long term. And so what we chose to do is actually look at this in the context of not just the percentage of DMSO, but also the cooling rate that is being used and the vehicle solution that is being used as well, because not many studies were looking at this combination. They were typically using a very sort of standard vehicle solution of perhaps FBS, and they were looking at a single cooling rate and then saying actually it's quite difficult to lessen the amount of DMSO we use because we see cell damage. And so from our studies where we tried both FBS, which as I said is a kind of gold standard in the research setting, to what we call plasma light A with some human serum albumin, and this is really what's used in the clinical context. And then we decided to try something called hypothermosol, which was a vehicle solution developed to enable hypothermic storage of cells and tissues, coming really from the transplant sort of community, and that's meant to help store at refrigeration temperatures cells and tissues. So we thought if we're going to do this kind of extended storage period in our DMSO, might that help as a vehicle? And so what we can see here is, again, we're looking just simply at membrane integrity via PI exclusion, and we're changing the volume of DMSO, the cooling rate, and then, of course, the vehicle solution as well. And you can see that hypothermosol was a disaster. We tried it, it didn't work. It was never meant to sort of be used as a freezing medium, so we can't blame the company. But you can see that really as soon as we try and drop the amount of DMSO that we use, we get quite a lot of membrane integrity lost, and it doesn't really matter if we change that cooling rate. Again, Plasma Light A actually worked quite nicely. We managed to retain, even by dropping down to 2% DMSO, above sort of 90% cell recovery, membrane integrity, at a range of cooling rates. Actually, the reason that cooling rate is really interesting is because it means that if you have slight differences between, one mechanical freezer compared to the next that you're using, that you would still have enough tolerance for your cells to be able to recover at slightly different freezing rates. But it also showed that, the work that we're doing matches what everyone says in the literature, because when we used FBS and dropped the DMSO rate, you can see that, or percentage even, you can see that we have a reduction in cell viability. But interestingly, that is at that 1 degree per centigrade per minute cooling rate, which again is typically what's used in the literature. But if you want to reduce the DMSO, we found that actually by increasing the cooling rate to 2 degrees centigrade per minute, you get your membrane integrity back to about that 90–95% level. So one of the potential reasons that in the literature, people tend to always say that you can't reduce the amount of DMSO beyond about 5% is because they tend to stick to a single cooling rate. So by using this sort of design of experiments approach, we're able to show that you can indeed reduce the amount of DMSO that you're using, but you may also need to alter the vehicle that you're using or the cooling rate, or both. One of the other challenges that we sort of put to ourselves is, if we're going to scale up our culture and we need to freeze clinically relevant cell numbers, realistically, we're also going to have to increase the density at which we store our cells. So typically within research laboratories, or if ever you tend to buy, cells from a commercial supplier, quite a standard sort of freezing sort of cell density is about 1 million cells per mil. And if you're working with cancer cells, for example, in a research setting, that's absolutely fine because they tend to recover quite well and they tend to grow so quickly that actually within a matter of a few days, you have quite a healthy number of cells to then work with for your experiments. But obviously, if we're going to put these cells into the patient, we don't necessarily want to infuse sort of 200 mils of solution. We want to infuse a smaller volume potentially. And also, if we want to, say, be able to seed the next scale of bioreactor, again, we want a higher concentration of cells in a relatively small volume. So we just sort of try to look at a small range of cell densities between 1 million and 10 million and show that actually you can scale up from where we are typically working at the 1 million, and you can quite comfortably, at least in terms of our cell viability, measure go up to 10 million cells per mil. Much more than that, and actually it becomes quite challenging to work with the solution because the cell, you end up with very little volume of solution compared to the sort of volume of cells. That becomes a little bit trickier to handle. But that was quite successful. We also found that in terms of, we're not seeing very much apoptosis, a slight trend perhaps 10 million, but nothing significant. And the cells are able to, grow nicely. When we looked at some quite simple measures of whether these cells are still stem-like and they are able to turn into sort of adipocytes, chondrocytes, etc., on the face of it, all looked very well. We just used different staining techniques. And here, this is just showing you the original cells and then them being stained for adipogenesis. So you can see the sort of oil globlets in stained red here. But what's interesting is that actually, although when you look at it, they all look fine, if you then lyse open the cells, collect the dye and do it sort of quantitatively, actually what we tend to see is that there are slight differences between each of the concentrations. So again, just giving us a hint that sometimes we need to be really careful about what measurement systems we're choosing to use. This was all the more important, really, because the work that we were doing in parallel was also looking at moving what we had been doing in microcarrier-based cultures in order to get these cells to grow in stir tank bioreactors. So microcarriers being small spheres about 200 micrometers in diameter, typically made of some form of plastic that the cells can grow on, and they are still therefore adherent, and they don't know much different to being in a flask, but we can have a really high surface area to volume ratio. But as part of moving into something that is more therapeutically relevant, we started to move away from serum-containing medium into serum-free medium. And what's really interesting is that actually we were noticing that cells are behaving differently depending on your medium choice. So, they were growing at slightly different sort of rates. You can see here a specific growth rate between a medium-containing FBS and serum-free, slight difference in cell size, which was interesting. And when we looked at images of our cells on these microcarriers, we were seeing just much more agglomeration sticking together of the cells in the serum-free measurement. And so we thought, actually, maybe what's going to affect our outcomes from cryopreservation is not just our cryopreservation protocol, but also even how we treat these cells upstream. And indeed, using that sort of standard 1 degree per minute freezing process and a serum-free commercial DMSO-containing solution, we can freeze the cells, we can recover them, pretty well. There's a slight drop compared to sort of normal passage, but not very much. But has changing that upstream culture condition changed the sensitivity of these cells to that cryopreservation sort of process parameters? And indeed it has. So we have here cells that were grown in DMM-FBS and then frozen in DMSO, and we get reasonable recovery, particularly at those sort of higher cooling rates. But when they've been grown in the serum-free medium, suddenly that picture changes. And as soon as you start to drop the concentration of DMSO, again, we see even larger changes in terms of how they respond to cooling rate sort of shifts as well. So just really interesting that changing that upstream process, which we thought, great, we're getting more cells in less time. Actually, if you then can't store them in order to get them to the patient, that's not going to help us so much. And again, we started to look at it with our sort of manufacturing minds in place and said, how does the timing of that downstream process affect cryopreservation success? So Tom and Andy had put together a sort of fake, if you like, manufacturing process that either took 5 hours or 7 hours, and a couple of different cell donors. So M2, which was on our very rapidly growing cells. So from a production perspective, you'd think, great, definitely need to use those. And then some that were really slow growing and they hated working with, if I'm honest, just because they were so sort of hard to get going. And so we kind of did this fake process whereby they are dissociated from the microcarriers, they're separated, concentrated, held in suspension. Then we add our DMSO containing formulations. We go through controlled rate freezing, we store, and then eventually thaw. And what it showed really is that in formulations that contain up to 5% DMSO, the membrane integrity was pretty much retained in all of our processes. But at 10%, as we're showing here, actually, we suddenly find that the longer our process is, so this is just immediately after the hold, but without any freezing or DMSO. And this is then if we have a 5-hour total process and 7-hour total process. At 7 hours, we start to see more of our cell population that wouldn't pass that 70% sort of minimum of FDA viability. Again, we start to see more aggregation as well in that longer process. And we start to see both that decrease in cell viability and increase in our cell aggregation more in our rapidly growing cell line than in our slow growing cell line. And again, even if the membrane integrity is retained, we start to see more of a drop in terms of how well they grow once you put them back onto plasticware, and how good they are at sort of forming colonies and being stem-like is really affected. And so Tom and Andy did a really big piece of work where they took lots of different cells that were frozen under these different conditions. Again, the blue being that really good high sort of growing cell line, and M3 being the really slow. cell line here in red. And if you look at sort of a nominal 50% attachment efficiency as being good, and that 70% membrane integrity as being your minimum cutoff for cell release, all of the red sort of triangles indicating the M3 cell line would pass pretty much that criteria. Perhaps a few here don't quite meet the attachment criteria, but you can see that many more of that blue population, the M2s, wouldn't. And so this shows that actually, a cell line that we might have discarded because it's slow growing and therefore potentially not very helpful as a production cell line, is potentially better at recovering post cryopreservation and surviving that process. A cell line we would have chosen for production because it's fast growing wouldn't. And so actually, if you have a choice within your allogeneic cell population, you need to think carefully about how it performs in different parts of your process, not just cell growth. We also started to ask ourselves, just as I'm getting towards the kind of wrap up, about whether we can even freeze cells on microcarriers. So thinking less about how we get these cells to patients, but how we can help that sort of seed train of bioreactors as we go through production. And the answer is that actually, yes, we can cryopreserve directly on microcarriers. And so what we're showing here is that if you normally transfer cells from, say, a T flask then into the microcarrier culture in a bioreactor, we normally get this sort of lag phase, a slight loss of cells as they're in that adaptation. Whereas if you inoculate a bioreactor with cells that are already on microcarriers, they perform really, really well. If you then look at actually those frozen microcarriers, and we did some SEM, you can see that they're retaining their morphology on these microcarriers even after that preservation and thawing process. And potentially this is what's causing some of that agglomeration that we saw in some of those earlier studies, because they do tend to try and collect together. But, you can see that basically we can retain, a really nice sort of profile in terms of how the cells grow if we store cells in these cryopreservation conditions on microcarriers, which is really quite exciting because it gets away from some of that initial expansion in T flasks as part of a production system. So what I've probably shown you, or hopefully I've shown you in these kind of, last 20, 25 minutes when I've talked about my own work, is that, we need to really look at how we design processes from end to end. So even if we take a process such as cryopreservation, which can happen at different parts of a bioprocess, it's really important that we think about, the impact that your choices we're making is having on that. And so, the decision to scale up or out is multifaceted, and it can't really be taken in isolation, because the fact that we chose to go for a scale-up approach and means that we hopefully for allogeneic therapies generate very large quantities of cells, then comes with challenges in terms of how we store them, the fact that we want high efficiency and perhaps for clinical reasons choose, for example, serum-free medium, has an impact on how our cells will survive through that cryopreservation process. So it's really important that we look at that holistic element. There's lots of people named here on the slide in terms of those that did a lot of the actual work that I presented today, and apologies to anyone that I have been missing off, and there are just some of our sponsors as well. Thank you very much for listening. Happy to take any questions.
Hello, Karen. Can you see me now? Yeah, okay. So once you, how many passage can you keep the cells going on without freezing? And then if you freeze them, will you retain the maximum number of passages without the cells entering into crisis?
So we've actually started to move away from talking about passage, and we start to talk about doublings.
Yes.
Because actually we find that traditionally we could get to about passage 8. But as I said, with serum-free medium, they are doubling more rapidly. So actually we were getting less passages, but finding we sort of reached a maximum number of doublings. Cryopreservation didn't seem to affect the maximum number that we got to before we hit some form of senescence. But I must admit, we haven't tried freezing at, say, passage 8 to see whether they will then, still be able to be pushed further, because that's probably not very representative of what would happen in the clinic and in companies.
Okay, I see. And what type of microcarriers do you use? These are solid, or you have porous microcarriers? And once you thaw them, will it the way you detach the cells from the microcarriers would be you won't have problems with this? They won't remain much strongly attached?
No, so we tried a range of different microcarriers, not necessarily with the cryopreservation project, but more generally. And a lot of our work was done with a Solihull plastic microcarrier. Although actually there was a range that worked really well, but they were all microporous, so the cells were firmly attached on the outside rather than penetrating. And we found that there wasn't any difference in terms of how the cells would detach from the microcarriers. We did a piece of work, particularly with the support of Alvini now, on looking at improving those detachment processes. We found that when we used the manufacturer's recommended sort of periods of time in terms of how long the cell should be exposed to trypsin or triple E, one of the sort of enzymes, we got a very poor recovery. So we found that actually we have to do a combination of time with enzyme, but also introducing some agitation in the stirred vessel in order to optimize that detachment. And that was the same before and after cryopreservation.
I see. So just one short one. When you talk about the cooling rate, you usually cool down to minus 70, 78 degrees centigrade, and then you go very fast in liquid nitrogen. So you're referring to that first stage, right?
Yes, referring to that first stage. So using cooled rate freezer rather than the sort of Mr. Frosty or Cool Cell device. Yeah.
Okay. Thank you.
No worries.
Anybody else? And I have a question, Karen, because I was not sure if what equipments are used in industry in order to produce these cells, because you told us that there are two alternatives with stir tanks and to scale up in the stir tanks, and in the robots or the machines. But
yes.
And it was not clear for me if it was cheaper to do it by the plates, automated by the robot, or instead of the stir tank. What is the situation in industry?
Stir tank is the cheaper option in terms of the surface that you need, or the space within a manufacturing setting that you would need. And because we can get obviously stir tank reactors of many varying sizes. So I think the maximum that's really been done in industry, to the best of my knowledge, is around 50 litres. And that gives us enough cells for a pretty decent sort of batch, given that most people are still at the sort of stage 2, stage 3 of clinical trials. The robot, however, is much better if you're looking at allogeneic therapies, because you want a separate, you want to be able to read those barcodes and separate one patient's cells from another, which would obviously be more difficult and mean you have to have lots of different bioreactor setups to do that. But a lot of work in industry now is also, and at the clinical sites, is looking at using some hollow fiber based bioreactors, and also looking at a all-in-one device, which is the it's called Milli something, like Clinimax, sorry, not Mini Milli, the Clinimax, which is supposed to be an all-in-one device, which can do both isolation, initial sort of grow out stage, and then some degree of cell expansion, and then be able to retain the cells back to the patient, all within one closed device. So that would be sort of a patient bedside type of system. I know that there have been some challenges in using that, particularly for adherent cells, but they've had more success with some of the non-adherent cell types.
Okay, thank you. Anybody else? No, I just one more final questions. When you how are the conditions that the cell should be in order to inject them to the patient? I mean, is there any protocol that you should follow in order to, I mean, in terms of temperature, of quantity, and all sorts of things?
There are some guidelines, but as I said, the sort of regulatory space is still really evolving, because we now have some cell therapies that are being used in clinic, particularly the CAR T cell therapies, and they actually are infusing relatively large volumes in a sort of blood bag type system. But I think it's probably a space that will change. So they are cryopreserved and they are thawed and then given to the patients, I believe at room temperature. But there has also been discussion, particularly when some of that DMSO work was first done, as to whether cells, rather than being thawed and given directly to the patient in an infusion, should be washed and allowed to go into some form of cell recovery for 24, 48 hours, and then the release testing done to make sure that we can give cells that are in the right sort of viable state to the patient. But that comes with challenges in terms of hospital settings having the right capacity to do that cell recovery and cell culture. So I think that's why, for the moment at least, where we're seeing success is that direct thaw and infusion.
Okay. Thank you very much. As there is no more questions, I would like to thank you very much, Karen, for accepting this talk and for giving this talk. And I look forward for collaborations with Loughborough in the future, and perhaps in the future you can come to Cuernavaca as well to visit us and to see our labs, and perhaps to talk more intensively with our researchers. Thank you very much.
Yeah.
Thank you.
Sure, there is another question.
Sorry, are you going to be at the ESAC meeting next week in Edinburgh?
No, but I hope you enjoy it. Edinburgh's a lovely city, and they've got a great lineup.
I didn't get your answer. You are or you're not?
I'm not, unfortunately.
So okay. Okay.
Thank you very much, Karen. Bye-bye.
Thank you. Enjoy the rest of
Thank
your day.
you. Bye.